All right, let's talk about the mechanism of how AMPA receptors get inserted, right? So the mechanism of AMPA receptor insertion. And so I'll start this little topic out by just saying that there are steps that we don't really recognize very well. And we think that there are going to be some intermediate players that we haven't yet identified, okay? So the first thing that has to happen is LTP gets triggered, right? So we induce LTP. And what that does, right, we've talked about, is allow NMDA receptors to sort of come on board. So allowing NMDA receptors to function. Right? Before they were non-functional because of the magnesium block, okay? And we know that this causes an increase in calcium. Okay, and we know that calcium works to be works as a second messenger, and it's through the activation of different kinases. So particularly we think PKC and CAMK2. Phosphorylate proteins that are necessary in inserting these AMPA receptors. So we'll just say help insert. And this is the part where, you know, there's some question marks around exactly what PKC and CAMK2 are targeting, but we know that AMPA receptors increase the number of AMPA receptors, or the density of AMPA receptors increase at the postsynaptic membrane here. So AMPA receptors onto postsynaptic membrane. Okay, and I think one of the major takeaway messages from this slide, right, is that these AMPA receptors are already present, right? So these AMPA receptors aren't being synthesized like de novo, that is brand new. So there's a pool of AMPA receptors that are already present. Okay, and somehow, right, because of the phosphorylation via these kinases, right, this pool of AMPA receptors gets inserted, and now you could imagine that the same glutamate that was here prior, right, before LTP, would activate these receptors and allow more, a stronger depolarization because more sodium would come in. Okay. Now you might say, but you said we could put on an antagonist to block NMDA receptors, and that we would still see LTP, and this diagram shows you need NMDA receptor activation to trigger AMPA receptor insertion. But the timing here matters. Remember, at least with the fluorescent experiment that we talked about in the last video, that they were doing that experiment like 30 minutes after inducing LTP? Well, so the NMDA receptor antagonist is put on 30 minutes after induction of LTP. So these AMPA receptors have already been inserted. So once the AMPA receptors are here, blocking this NMDA receptor doesn't really have much of effect, has really any effect, on the activity of the receptors that have already been inserted and can now pass sodium current. Okay, and so this AMPA receptor insertion mechanism is great for, you know, that hour or 2 hours or 3 hours where LTP is seen, but there are occasions where LTP can last even longer than that, okay? And so this next point of discussion has to do with this idea of making new AMPA receptors, right? And so in this figure, the AMPA receptors are always present. That's why I said that's an important point, not always, already present. Okay, so we have some pool of AMPA receptors that are ready to be inserted. But there's also a possibility that new AMPA receptors could be synthesized, and that would lead to the really prolonged type of LTP that's seen. So let's look at that. So let's look at prolonged LTP, right, on the magnitude of many, many hours, even days. And what's going to be happening here are synthesis of new receptors. So AMPA receptors especially, but also NMDA receptors. As, and this is an important point too, the dendritic spines may grow. And so we've seen early phase LTP, or what they call initial phase LTP here, right? So the first mechanism would be increasing calcium coming in here, right? And this is through NMDA receptors, and we've talked about how that happens. We mentioned that this increasing calcium can lead to increasing AMPA receptors, right? So not pictured here are insertion of AMPA receptors. But we think of this, like I said, as this early phase, this initial phase that's going to go from minutes to hours. But now we want to talk days. Long-term or late phase is a better term, right? So late phase LTP requires changes in protein expression such that we're going to make new proteins. Okay? So the way that this works, as I mentioned here, right, that increased calcium can activate different kinases in this initial or early phase LTP, right? So we talked about, PKC, CAM kinase 2. Another one of these kinases that can be activated is PKA, right, through increasing levels of calcium and different intracellular messaging pathways. We see that here's the enzyme adenylyl cyclase producing cyclic AMP, and the result is protein kinase A activation. So with PKA activation, right, transcription factors get activated, so this leads to things like leads to CREB, right, a transcription factor we've talked about before, being phosphorylated. Right? And so in the diagram here, you can see that we've got a transcription factor CREB that's been phosphorylated as pCREB, or phosphorylated CREB. And this increases a whole bunch of proteins expression. So one thing that it increases are those receptors. AMPA receptor expression goes way up. NMDA receptor expression goes up. There are other proteins that are involved in growth of dendritic spines. So synaptic growth proteins go up. Okay. What we think this does is actually form new dendritic spines that weren't here previously. Okay. So this entire spine might be a result of induction of LTP. And so because this new spine is here, we're also going to insert the requisite proteins, right? So there's AMPA receptors here and there's NMDA receptors here. And so there are entirely new points of synaptic contact form. So it's not just about more current flowing through AMPA and NMDA receptors, although that'll be a result, right, because they're existing here and here. But there are these entirely new points of contact, new points of pre- and post-synaptic contact. Right, between the pre- and post-synaptic cells. Evidence for this occurs when we give protein synthesis inhibitors in the bath of these cells. So here's the experiment, right, where we're looking at the postsynaptic cell, right? So we're looking at this. Let me clear up some of what I've scribbled on here. And so we're looking at what's occurring in this cell here. And if they stimulate the CA3, they're going to measure a response in CA1, okay? And whatever those responses are, that's going to be 100%. So whatever that EPSP, maybe that's 5 millivolts, okay? That 5 millivolts is considered 100%. So we stimulate CA3, measure the response in CA1, that response is 100%. After LTP is induced here, that's what the red arrow is the induction of LTP via high-frequency stimulation. Now, as you would expect, when they stimulate the CA3, they see, right, this facilitation. And it's quite extensive. We're talking 8 hours after LTP was induced. These synapses are responding, and the postsynaptic cell is responding with like 175%, like almost twice the magnitude of an EPSP. But look what happens here when they add their protein synthesis inhibitor at the same time as they induce LTP. They do see some initial LTP, right? So you go from 100% upwards to 200%, but it fails to be prolonged, right? So we think that this initial phase, right, this is still initial phase, and it's still working fine, but there's very little or much reduced late phase because of that protein synthesis inhibitor, right? And so maybe that's worth noting, that this late phase, LTP inhibited when protein synthesis is blocked. Okay. So, maybe one more thing to look at with respect to LTP induction. And the book mentions it, but there's not a figure of it. So I'm going to back up and erase some of the things that we had here. One of the other mechanisms that the book mentions, potentially, is the idea that there can be retrograde messaging. So the idea here is that if a neurotransmitter from the presynaptic terminal is co-released, right, so maybe there's a release of something like nitric oxide from the presynaptic terminal. Okay, so if there's nitric oxide here, so co-release of another neurotransmitter. And so I'm just using nitric oxide as an example. It's the example that the book uses, right? That nitric oxide could be released. It doesn't need to be released from a vesicle, right? As it's a gas, so it can diffuse out and bind to receptors on this presynaptic membrane, right? So there might be receptors for nitric oxide out here. So co-release of another neurotransmitter activates presynaptic receptors. These presynaptic receptors eventually lead to a transient rise in calcium. So this presynaptic calcium goes up. And having more presynaptic calcium around increases the amount of neurotransmitter released. Right? So with more calcium here, when this presynaptic neuron is stimulated, calcium comes in, but there's already calcium present here. Okay? And so more calcium equals more glutamate released. Okay, so this wouldn't be one of those late phase mechanisms of LTP, but more of an early phase mechanism. That would trigger larger responses, right, in this postsynaptic cell, because there'd be more chemical to bind to AMPA and or NMDA. Okay, I think that's good for LTP. I'll come back and talk a little bit more about LTD next, long-term depression.